Good morning. Hello. There is one question left. I don't know whether you tried this question. Was very beginning. We have these questions like two tutorials. So now we took almost all question from these tutorials. This should be the last question. Uh, did you try this or not?
Yes, sir. I tried it.
Okay. Do you have answer now?
Uh,
I think the first word is missing. It could be this word, Karen and Steve. Each have a sibling with sickle cell disease. Neither Karen nor Steve nor any of their parents have the disease. And none of them have been tested to see if they have the sickle cell trait. Okay. That means they haven't tested. None of them have been tested to see if they have a sickle cell trait. I think we discussed sickle cell disease last week under the concept of pleiotropy. So pleiotropy is conditions in one gene, but which can affect to multiple traits or phenotypes. So sickle cell is an example of the pleiotropic conditions. There is a single change of a gene, but people show a widespread effect in their body. It's not a one place. So many places are affected with sickle cell disease. So they haven't tested to have a sickle cell trait. A sickle cell trait. This word explain a conditions of the genotype. Like what is the genotype in sickle cell trait? So if sickle cell condition is capital S, like hemoglobin S. Usually we write like this, especially for sickle cell disease. And healthy one, we can write example like this. So this is disease. This is healthy conditions. Now, if it is come like this, the homozygous recessive form, they have a sickle cell disease. Then the next combination is healthy and disease conditions. So this is a heterozygous. So homozygous recessive, this one is heterozygous. So heterozygous is sickle cell trait. And the next one is completely healthy. Sickle cell disease. Sickle cell trait and healthy. So calculate the probability that if this couple, the ability will have sickle cell disease. Okay, this is also considered as a carriers, the carrier stage. So they are the carriers. They don't have the disease, but when two heterozygous couple married, then they can have a child with a sickle cell disease. Calculate the probability that this couple has a child, the child will have sickle cell disease.
Sir,
Yeah.
sir, can we take one as YY XX as well for the one person XY, like then put the things?
One thing is sickle cell disease is not a X-linked disease. Since it is not X-linked disease, we try to avoid taking X or Y. But this is something like we write a standard way of writing, and we don't take X chromosome or Y chromosome, but HB indicate hemoglobin. For example, we can write this as a capital A or simple A. So this is mostly we use for Mendel genetics, and when we use a non-Mendel genetic, example X-linked disease, we write X hemophilia like H and Y like this. Now this is clear. Mendel genetic we write like this, and X-linked disease we write X chromosome and Y chromosome. And also we have conditions. We learn incomplete dominance and codominance. When we learn incomplete dominance and codominance, we used to write like this. If it is a color, capital C, red color, color, white color. So this is another way of writing. So we have these all three type of basic standard of making the genotypes. So this is the Mendel. These two are non-Mendel. This is X-linked, and this is incomplete dominance and codominance. So if we consider sickle cell disease, it is also some kind of codominance situation, because this sickle cell trait, now look at it here, it's a kind of the way we write, instead of giving C, we write HB, it's hemoglobin. So hemoglobin A and hemoglobin S. So this is very similar to codominance standard of genotype writing. So this has a little bit of a scientific background, because sometimes the sickle cell trait, they also express the disease to different level. They are not completely unhealthy, but they can also express certain level the condition. That means the S, this one is the, this is causing the disease. Having one allele, it can also affect these healthy conditions. But we don't consider, I mean, in genetics textbook, they don't consider this is completely codominance or incomplete dominance. But when we write like this, we can write like a codominance or incomplete dominance pattern. And there is a situation, actually simple S recessive one, also certain degree express. That means both conditions showing. Just like a codominant situation, somehow it is not considered as codominance, but when we write sickle cell disease, we write like this. So now we can make a conclusion. This is the basic Mendel genotype writing. This is the X-linked writing, and this is a codominance. Since sickle cell is a little bit somehow similar to a codominance conditions, we used to write like this. So this is the explanation why we not include X and Y, because it's not sex linked. So we like to go towards the codominance conditions, because heterozygous condition, there is certain degree of expression of the disease. Okay, so we try now what will happen if probability if a couple married with a sickle cell trait. So hemoglobin A is S. Okay. Now we can understand now. One person can develop the disease. So if coupled with these heterozygous conditions, if they marry and there's a chance to getting a probability is 1 out of 4, the disease conditions, right? Now, according to this question, what we can understand, uh, they have a sibling with a sickle cell disease. So when they have a sibling with sickle cell disease, this condition is already arised. So then we have to consider now rest of the situation, these three, because we have to exclude this because it is already there. One has a sickle cell disease in the family. So then he already uh, showed this one. Then the next member in the family has no sickle cell disease, but then it should be come under this box. So then the chances of being a carrier is this, probability is this, because 3 out of 2 they have a sickle cell trait. I mean, this should be A and S. Okay, A and S, A and S. So these two, this one and this one, there is a chance of become 2 out of 3. Right now, in both condition, that's the same. That means this is the Karen and that's a Steve. It's the same for them. So Karen and Steve exactly same because in their family, again they have a sibling with the sickle cell disease, and Steve again coming out of 3, uh, the probability is 2. Right now, this is the probability of being a carrier. Now, next thing is question is asking if they marry and then they have a child, what is the probability of getting sickle cell disease? So if we consider that as a separate event, so when they marry and having a child, so the child has a chance of getting sickle cell disease this much. Because carriers can make a probability of having one child out of 4. Now we have uh, 3 probabilities. So what we can do, we can apply the law of multiplication. So like this. So 3 times 3 is 9, 9 times 4 is 36. 2 times 2 is 4. So this should be simplified 1, 9. Okay, this is the probability now. 1 out of 9 is the probability of having a child with sickle cell disease if they are carriers. And we have to exclude the special situation here because they already have a one member in sickle cell disease.
Uh,
So this is the
so,
solution. Yeah.
what's the last questions in this? Like they asked.
This is the question, right? The last one in the first tute which I sent. Check it. I think this is the last question.
Yes.
I think second one is past exam question, right?
Yes. Second it is all past
Yeah,
exams.
all past exam questions. It's not all, but we have another tute coming up because we learn non-Mendel genetic. Some of the still question left from non-Mendel genetic. So this is not all questions. Still some question we can find from non-Mendel genetic. I think if we get all these other rest left question, then maybe we have done all question, almost all MCQs from the past papers, like from 2000, I think 9 around, even before. Some questions are even before.
So, they are saying to calculate the probability of a couple which has sickle cell, no, sir?
Are you asking this question?
Yes, sir.
Yes. Now the Steve and Karen, they're going to marry and they're going to have children. So what is the probability of a child develop sickle cell disease is the question. And the question is quite, easy if they ask like this. So Steve and Karen are both are carriers. What is the probability of having sickle cell disease? So then it's a 1/4. So it's the easiest question. One is carrier, other one is carrier, and when they make a child, what is the probability? So it is come under this category. That means the first thing we discuss, easiest one because it's a 1/4. But this question, they have provided additional information. Already their families, Steve has one sickle cell person in their family. Karen also have one sickle cell person, the siblings. And only that fact we have to consider. So they have to exclude that one because already there. Then mean the chances become a carrier is this. So we consider this is the chances to become a carrier. Otherwise, to become a carrier, if they don't give this sibling like these conditions, is a half. Half is the carrier. One half is the carrier to become sickle cell disease. If we don't have this special factor, one member has a sickle cell disease. That means question is easy, but question is bit difficult. We have to exclude this one now and getting three now. Three chances. There could be two. So that's why both families we take these two, and then if they have a child, that's obvious. That is 1 out of 4 to become a disease, sickle cell disease. So we all multiply to get the final answer of this.
Uh, so two-third in both the families are for the carriers and healthy, right?
Yeah, we also include healthy because uh, if you look, this is all the probabilities. There are 4 probabilities. If the carriers they want to make a children, then the probabilities, all probabilities are there. One is healthy, and two carriers, and one is a disease. So what we did when they have already disease one, we exclude that one because it's already there. It happened already. So then we take these three probabilities. So because this has happened, they have already won with the sickle cell disease. So we exclude that, and there is now three chances. One is healthy, two become carriers. So then the carriers in this family is two out of three. So it's the same with the other family. So then, based on that, we get the final answer.
Oh. So if we multiply those both, won't we get the this thing for healthy? The healthy and carrier, two-third into two-third.
Two thirds are carriers, right? Because these
Yeah.
two third are carriers. That means when we are multiplying the carriers only, the carrier stage. So then that is we consider only carriers. We are not considering the healthy one. So only carriers we are multiplying. The probabilities is 9 out of 4. And separately we consider if they have a child, this is a probability. Then also we multiply that one to get the final number.
Okay, sir, got it.
I think we discussed last week the sickle cell anemia. So this is an example of pleiotropy. There is a mutation in the hemoglobin gene which can affect the person different level. So sickle cell is a shape of the hemoglobin. So it become like this. Instead of the biconcave shape of hemoglobin, change to a sickle cell. And anemia is lack of blood. Right now, why I think we haven't discussed too much, right? We just introduced what is pleiotropy. Pleiotropy is defect in one gene, but effect in multiple places of the body. So we only uh, just discuss what is sickle cell anemia. So it is basically the due to the shape of the red blood cell, the person develop a lack of blood. It's a amount of hemoglobin and the blood level decrease. Right? I think that's what we discuss, isn't it?
Yes.
Yes. Now, do you know why this red blood cell has this biconcave shape? What is the reason to have biconcave shape? So it
Absence
means
of new.
like, I mean, 3D shape, if you look at it, I don't have 3D picture, but if you take 3D shape in the middle, there is some kind of a embedding middle. It's embed a little bit down to this inside, to the center. So, and if we look like this way, it's a biconcave shape. Do you know why this shape is important for red blood cells?
It increases the surface area for the oxygen to come by.
Yeah, surface area. So why surface area is important? When we consider the main function of hemoglobin is to carry oxygen. So main function is to carry oxygen. And how oxygen cross the cell membrane is diffusion, simple diffusion. So, what is simple diffusion? Movement of molecule from high concentration to low concentration. If oxygen level is high, oxygen can move into the hemoglobin and it is cross the lipid bilayer. Oxygen can cross the lipid bilayer because there is no restriction from lipid bilayer to cross the oxygen. And when we look, what are the factors affect diffusion? There are several factors are important for diffusion. One is the surface area. Surface area is one factor. Second factor is the diffusion distance. Third one is a concentration gradient. So concentration gradient is if there is oxygen is very high in one side, other side is very low. then it's faster, like this. So here the gradient is, if this side is very high, this side is very low, then it's faster. And the other one is a temperature. So these are the four factors which affect the diffusion. This is something it's very important in biology, not in this place, but many places. The diffusion is one of the important movement. So what are the factors affecting diffusion? Surface area, diffusion distance, and concentration gradient and temperature. So surface area increased by having this shape. Now what will happen if the hemoglobin molecules, they are like this. So hemoglobin molecule consists of four subunits. One is alpha, one is beta, and one is beta. So hemoglobin is a protein. So this protein is synthesized by the cells as what we discuss, normal transcription and translation mechanism. That means the gene coding for hemoglobin responsible to make the messenger RNA, and messenger RNA produce protein, the hemoglobin subunit. Now, if there is a defect in this sequence of the genes, if there is a nucleotide change, then there is a chance of this amino acid change, because this is a polypeptide chain which is made out of this amino acid. So when you're learning molecular biology, I think we can understand every single step, but basically what we can still understand, gene is a specific sequence of a DNA molecule responsible for polypeptide chain or proteins and character. So this is a hemoglobin gene, and this is a polypeptide of the subunit. So if there is a change in the sequence, then what will happen? This amino acid change. If we get a strange amino acid which is not supposed to be there, then the, the complete shape is changing, because amino acid, order of the amino acid important to decide the tertiary structure. So primary structure is the sequence of amino acid. Secondary structure is the hydrogen bonds, beta pleated sheet and alpha helix. Tertiary structure is the 3D shape of the protein. That is the most abundant and most functional protein is 3D proteins. So in sickle cell disease, there's a mutation in the gene. As a result, they produced a hemoglobin. It is not supposed to be like this. The hemoglobin is quite a strange shape like this. So they make instead of the globular round shape hemoglobin, they make a long shape hemoglobin. So this long shape hemoglobin cannot pack in red blood cells to make the biconcave shape. They have to organize like this in the red blood cell. Surprisingly, there are 270 million hemoglobin molecule in one cell. So 270 million hemoglobin molecule in one cell. Now you can understand, if the 270 molecule going to pack, if they have a different shape, they make completely different shape of hemoglobin. So the shape is like this. So when the shape is like this, it lost the biconcave shape, and it is cannot move easily in the capillaries, because if you take a capillary, this is fit enough to one cell. So this cell nicely fitting to capillary. But if we take a sickle cell person, there are several going to pack in the capillary. What will happen if several going to pack in the capillaries? They can easily block the capillaries. It's called clogging. When capillaries are clogged due to the pressure, they can break down. The hemoglobin can, red blood cell can break down, and even it can start bleeding there because of the break in the capillaries. The blood can go out, so it start bleeding. So this is the problem with the sickle cell disease. Uh, how we can now understand the output if bleeding happen, organ does not get enough blood. Organ does not get enough blood. Organ does not get enough oxygen and nutrients. Organ does not get enough oxygen and nutrients. The cells are not properly functioning. So cells are very unhealthy. As a result, for long term, the organ dysfunction happen. Organ are not functioning properly. If organ dysfunction happen, organ failure can happen. So kidney failure, heart failure, liver failure, these are the failure of the most of the important organs. So these people develop tissue damage, bleeding, organ failure, and also, do you know this word thrombosis?
Yes.
So thrombosis is
Okay.
formation of blood clot inside the blood vessels. So this clot can travel with the blood, and it can block different places, like even a heart. If they can block the coronary artery, so they can develop heart failure. So heart muscles are dying because of the blocking. Later, they can develop heart failure, and also they can block capillaries of the kidney. So when they block capillaries of kidney, kidney filtration affected, they can develop kidney failure. So finally, single gene which can affect many places in the body. This is an example of a pleiotropy. So any questions about this?
So the surface area affects simple diffusion. Uh, so is it the surface area of the red blood cell or the villi cell sort of thing?
Red blood cell of the surface area of red blood cell or
The villi, no, the alveolar sacs.
alveoli.
Yeah.
Uh, no, there are situations alveoli is also affected because if we consider lung oxygen diffusion, then first the alveoli, if this is just example alveoli surface area is also important because when we inhale, oxygen come into the alveoli. Then if we take a capillary, so capillaries, let's consider this one is a capillary, and this one is the red blood cell. So this membrane is also important, and then it passes through the capillary membranes, and then it passes to the red blood cell. Then how many membrane? It cross? So it cross actually uh, the alveolar endothelial cells. So alveolar endothelial cell has two membranes because this side of the cell and this side of the cells. And capillary alveolar epithelial cell and the capillary has capillary endothelial cells. So then it can also pass this 1, 2, 3, 4, 5. So it crossed 5 membranes. So when we think about these 5 membranes, if alveolar surface area is one of the important thing, oxygen should diffuse. If the capillaries are less, or capillary is high, or capillary is not affected, then the next one is red blood cell membrane. So let's assume capillaries are constant, they are not changing. What other things can be changed? Alveolar epithelial surface area can change. Why alveolar epithelial surface area change? Due to disease conditions, they lost alveoli. If they lost alveoli surface area, then this diffusion is affected. The person has a completely correct red blood cell is fine, and person is having a alveoli, everything is perfectly okay. So